In other words, chapter 28, the reproductive system, is going to produce a real contrast to the 10 systems that have preceded it. Because this is a system that actually differentiates human beings into two kinds, male and female, to address a distinct reproductive role. That will mean literally that different organs are activated and that different roles in reproduction are fulfilled. It's interesting that the reproductive system is the only one that is not essential to our life. If you remove any of the previous 10 or compromise them, and by that I mean go beyond wounding. I'll grant you that the integumentary system, I can get a cut. That will heal up. However, if I remove the integument, you die. And that's true of all the other 9 systems as well. But in the reproductive system, you can remove the functional organ of the reproductive system, the gonad, from male or females, and it does not kill. In fact, you can live a long and healthy life without your reproductive organ. Now, that long and healthy life has been recognized not only in humans, but in many mammals. In fact, we use this as a strategy in the production of our animal products. So leather, meat, and the chemicals that we get from bone or from skin or from blood, all of those things that we harvest when we raise an animal for human use, does not require the reproductive system. However, the reproductive system does have a profound effect on other organ systems. So after a fairly comparable and fairly neutral adolescence, we reach puberty, and the reproductive system differentiates our body to display and perform the reproductive role that we carry around individually. 2 genders and 2 distinct and different roles. The female incubates the developing embryo, hosts its fertilization, incubates the embryo, and delivers the living offspring. The male and female reproductive organs produce and store specialized reproductive cells that have to combine to get a new individual. Now, we know we must do sexual reproduction if we are reproducing as a human. We are obligated to that type of reproduction, and that's unusual. If you kind of look at the whole scope of biology, there are so many organisms, from single cells all the way up to blue whales, where you have options in reproduction, meaning that you can reproduce asexually, you can get a new individual just by cell division, or if you're multicellular, you can reproduce by clonal reproduction. Plants are well known for this. If you cut off a branch and stick it in water, some plants will develop roots. You plant that, you've got a new individual. So that asexual is complemented in those plants by the ability to do sexual reproduction. But although there are plants that reproduce by outcrossing, 2 individuals contribute to the offspring, there are many plants that can reproduce by pollinating themselves. So it's sexual reproduction by one individual. There's lots of organisms that have retained that versatility in terms of reproduction. Human beings, in fact, all animals in the animal kingdom are, that's less well known. And most animals reproduce obligately by sexual reproduction. That means something very important to me as an evolutionary biologist. If it is that universal, then there must be some big advantage to it. And I want to tell you what that is right now. The advantage that sex has is that it generates variety among the individuals in a population. Everybody is genetically different. It turns out that in a changing environment, both short-term and long-term, genetic variety in the population is a big advantage. We'll return to that point again. In order to accomplish this, we're going to have to activate glands, and those glands secrete sex hormones that guide the differentiation of our reproductive organs. The continued secretion of those hormones are going to maintain those organs, are going to monitor and activate the production of gametes, their storage, are going to regulate our behavior in mating, our mating behavior, so that we accomplish mating and fertilization, and that the species continues. So that's really quite a few things for one system to do. We're going to look first at the organ systems, as shown in chapter 28. The idea of the organ systems is differentiation, male and female organs being different. Now, we are raised that males and females are very different. There's absolutely no question about our gender identity being a big part of our upbringing. But I kind of want to point out that the actual genes are largely shared. Both males and females have X chromosomes. So all the genes on the X chromosome are shared by both genders. The number of copies produces some interesting differences in their expression, called sex linkage. The Y chromosome is the chromosome that differentiates a human body into a male body. So we're going to see that as actually a very reduced chromosome compared to the X chromosome. It's a chromosome that will produce a male body by carrying genes that largely relate to male reproductive organs and male fertilities. The pathway for the male. I want to first give you a term, gonads. Gonads refers to a sex organ. It applies both to the testes in male and to the ovary in females. Both are called gonads as a general term. In the male, the testis is the male gamete. This is where the male organ, this is where the gamete is produced. Now, all living things are made of cells. All cells come from pre-existing cells. But this is the first and most prominent example where we're actually going to take the body cells and we are going to make a different kind of cell, a reproductive cell. It is going to be not different in minor ways. I will grant you that a red blood cell throws away its nucleus and packs itself with hemoglobin. That's absolutely true. But it starts out as a normal diploid cell. It is the expression of genes to make a red blood cell that make that red blood cell different. Whereas in this case, we're going to make a gamete that is reduced in the chromosome number, but not randomly. It's going to be a cell that has an exact half set of the chromosomes. It's not just good enough to have 23 chromosomes. You have to have a set of chromosomes, a complete genome. And the sperm in the egg must be fulfilled and competent in order to get normal fertilization and a normal offspring. Now, I want to point out that some forms of life are really, really tolerant of changes. So you'll get a multiplication of chromosomes, so you might be carrying an extra chromosome or you may be missing one in a plant or in a microorganism that looks completely normal to us. Not so in animals. Changing a chromosome number always brings a noticeable effect. From the testes, where the gametes sperm are produced, we store them in a collection vessel called an epididymis and prepare them for movement. Now, movement out of that is episodic. The sperm is collected. We want the tank to be full until a mating event. And at that point, the event of climax and ejaculation expels those sperms along the tract. that go from the epididymis to the ductus deferens, the ejaculatory duct, and the urethra, the pathway out of the male body. Now, along the way, the sperm and its fluid are changed into semen by adding glandular secretions along that pathway. We'll look at that when we take a look at the male genitalia. Along the way, these accessory organs increase, are preparing these fluids. The seminal glands, the prostate glands, and the bulbourethral glands are each producing separate products that turn the sperm fluid into semen. The semen is a more complex fluid that has supplemented the sperm, the living, swimming male reproductive cell, with fluids that will prolong its activity, will support its survival, and support the function of it reaching the egg for fertilization. But I kind of want to point out what a really low probability that is. There's huge selection pressures on sperm. A normal sexual congress may lead to the deposit of 200 million sperm in a normal ejaculate. If 50 sperm reach the ovary and are in a position to fertilize if ovulation occurs, that is excellent sperm performance. So I don't even want to express that as a percent. 50, or if you think about both ovaries, 100 sperm out of 200 million, that means most of those sperm die along the way. Now, externally, we see a fleshy sac called the scrotum that holds the testes out, enclosing the testes and protecting them, but also holding them separate from the lower pelvic region of the body. Suspending them in the scrotum lowers the temperature noticeably, and that's an important quality. The fact that all mammals have these testes external to the body shows the importance of controlling the temperature, and by that I mean cooling the testes. This pathway that we just talked about goes out through the urethra, goes out through the penis. This is an erectile organ, and the urethra there is as we've mentioned before, a urogenital organ, performing the function of urine excretion on a daily basis, multiple times, and performing the function of sperm delivery during sexual episodes. So we have seen this before, where the testes are located here in the scrotum, a descending sac, and generally it also separates the penile organ from the anal outlet. I mention that specifically because it's going to be important later. And from the testes, gametes that are produced mature into spermatozoa and are stored here in this arc, the epididymis. They await an episode of sexual excitation and erection and sexual congress. When ejaculation occurs, the pathway of the spermatozoa is through this ductus deferens, sometimes called the vas deferens. And notice its kind of convoluted path, ascending up and over the pubic symphysis, over that pubic bone, basically rising above the urinary bladder. And I believe, I get the back half of this. You can see how it circles around, basically passing behind, posterior to the ureter, and then descending here along this spinal and rectal region of the pelvic cavity. Now here is the seminal gland. Here is the prostate gland. And finally, unfortunately, this figure split the bulbourethral gland. I'm going to go back. Here is the seminal and its addition. Now you can see that this upper tract is delivering the sperm, and the seminal gland supplements it. As you come down here through the prostate gland adds fluid, and then the junction with the urinary tract occurs here, and down further into the urethra is where the bulbourethral gland joins. So from this point to this point is where you're turning sperm and fluid to semen. After that point, it is muscular contractions in the epididymis that are propelling the sperm through this tube, and the pathway leads out the urethra to the tip, the external urethral orifice at the tip of the penis. So that's the pathway. When we look at development, we're always fond of this. We start out as a single squishy cell, and we're going to look at how that develops into an embryo and implants and so forth. But right now we're just doing the organs in the male body. You can kind of see the pelvic cavity shown here. You can see that the peritoneum and the testes development starts high on what will become the retroperitoneal space of the abdomen. And through development, so from 2 months, you'll notice it's still perched high on that posterior wall. It actually descends as that tube shortens, pulling it down to a position in the in the pelvic cavity right above the base of the penis. Now, as development continues, by birth, this testis should descend through this pathway into the scrotum and be obviously located there from the outside. Undescended testicles are not a common, but a kind of not an unusual characteristic in humans and in other mammals, a recessive trait. Often these undescended testes simply correct themselves as time goes on. The accessory structures show how the testis here is maintained and supplied. The visceral layer of a membrane, the tunica vaginalis, covers it. And you'll notice here the so-called spermatic cord. Now, the spermatic cord is responsible for bringing blood supply, which you can see here, arteries and veins descending behind the testes, and the ductus deferens is shown next to it. Another artery is shown here, and the genitofemoral nerve is providing the nerve services that monitor everything, sperm production to a sexual event. You'll notice the spermatic cord sort of forming, it's not formally called a plexus, but it does look like a braid of tubes. Look at all the tubes that are necessary in order to reach this external position. You do notice that there are various layers, the outer skin lying above the parietal and visceral layers of the membranes that actually hold the testis in place. Following the ductus deferens up behind, you can see the urinary tube here to the back of the bladder and its previous, as was shown in the previous slide. Now, male reproductive function is affected by temperature, and the temperature differences are not profound. 1.1 degrees C or 2 degrees Fahrenheit is the normal cooling that is achieved by this external and descended position. One strategy that's used to address male infertility is to suggest that the testes be less confined by underwear, so boxer shorts are recommended in an effort to lower that temperature. Just a few degrees makes a big difference in terms of sperm motility and sperm longevity. In other words, how fast do you move the flagella and how long do you move the flagella? Muscles do contract in order to regulate the level of the testes. So in warm weather, they relax and the testes move down and away from the body. In cold weather, the testes are pulled up to the very wall of the pelvic cavity to maintain the consistent temperatures. Now let's kind of talk about cell division in a review sense. We talked about two processes, mitosis and meiosis. And they involve similar mechanisms and similar organelles to accomplish the division of the chromosomes in the nucleus. By the way, cell division consists of mitosis and cytokinesis, or meiosis and cytokinesis. The division of the cytoplasm, the separation into different compartments, is not really what we call mitosis. Mitosis is the division of the nucleus. I want to first deal with the simpler and more universal process because this is the process that supports our growth. We start out as a single squishy cell. That single squishy cell is divided many, many, many times to produce our trillions of cells that exist throughout our lifetime. And then those general products, what we would call sister cells, that have gone through mitosis. So you had one, as shown in the figure, now you have two. They're exact genetic copies of the original cell. Those sister cells are typically going to be smaller. They're then going to grow and divide again. So from the fertilized egg, we're going to have 2 cells, 4 cells, 8 cells, 16 cells, and on up as mitosis races forward. This is an equational division. The copies are exact, and they all copy the original fertilized egg. That's because of DNA being exact in its replication, and the division then of the 2 copies of every chromosome being exact, sending 1 copy to one end and 1 copy to the other end. So the first key to recognize in this process that supports our growth is that you start out with chromosomes of a characteristic number and relationship. We have 46, and those 46 chromosomes are actually 23 pairs. We number most of them in order 1 through 22 pairs, so 44 of the chromosomes are what we call autosomes. And then we have a pair called the sex chromosomes. 2 X chromosomes in females and X and a Y in males. But the balance of that set is extremely important. Now, what's drawn in this figure are just 6 chromosomes, because we could repeat this for 46 chromosomes, but it would just be confusing to look at. So what we've done here is we have a pair of chromosomes shown here. Notice how they're morphologically the same length, the same position of this constriction called the centromere. And also they are different shades of gray. Gray and black indicate paired chromosomes. Now these 2 chromosomes represent a chromosome that has the same genes in the same order from end to end. They also represent the contribution of the different pairing. If the black chromosome came from the father, then the gray chromosome came from the mother. That's how we got a pair. We're going to later show meiosis is not equational. It produces very different products. So here we have, let's call it the red set with a dark red and a light red. Here we have the blue set with a dark blue and a light blue. And the first step that's important in mitosis is DNA replication. Now, we saw complementary base pairing, and we saw the nucleotide base sequence as those critical factors that are on the molecular level that enforce the exact copying and distribution of DNA materials in chromosomes. So what do we see down here? Each chromosome has been replicated end to end. So on the black chromosome, I'm going to draw down one chromatid through the central constriction. The other one is drawn opposite it. But these are basically two exact copies of the same chromosome. You can see that each of the chromosomes, there still are 6. Chromosome means colored bodies, so there are 6 colored bodies here from the staining you do when you prepare the cell. However, you'll notice each one of them is double-stranded. Now, in order to accomplish mitosis, we're going to line them up on a central plate, and for each of these centromeres, we're going to pull one in one direction, and we're going to pull the other strand in the other direction. So black and gray do not lie next to each other when we line them up, but the blacks separate with one strand going each way, the grays separate with one strand each way. We continue on down the line, and after the separation has occurred, look at what we have. Daughter cell number 1 looks like this. Daughter cell number 2 looks like this. And if you compare, this daughter cell is exactly the same as the original parent cell. The same thing applies to daughter cell number 1. So, labels in your mind to hang on this topic of mitosis. Mitosis is cell division. It is equational. The copies of the DNA, the genes, and the chromosomes are exact, and the division is precise to produce exact copies. Now, after we produce 2 cells from the fertilized egg, and then from there go to 4 and 8 and 16 and 32, and we just keep doubling that number, the cells start to express themselves genetically so that the growing embryo, which after implantation transitions to a stage we call the fetus. It goes from a ball of cells that basically looks, well, the cells are uniform, to a developing body of what in time clearly becomes a human being and a human embryo. Those cells are differentiating. So in a particular place in the embryo, it's going to form eyes. In another place, it's going to form arms or legs. That process is called cell differentiation. Cell differentiation is the expression of the genes in the nucleus that say, make an arm here, and equip it with all the muscles, nerves, skin, and bone that are needed to make an arm. Now, an interesting thing, this cell right here in my finger, it has all the genes to make eyes. It has all the genes to make an ankle or to make a liver. But those genes are not expressed in that skin cell. What's expressed are the set of genes that say, make an integumentary cell, in this case that outer surface cell of the epidermis. It would be an epithelial cell. It's forming part of the integument. So it's the differential expression of genes that produces our different tissues, organs, organ systems, and also the expression of the genes and the proteins they produce, working as enzymes, working as cofactors, working as components like hemoglobin or collagen that provide the structure and also the regulation of the metabolism. So that's how we're growing. So far, all we've done is grow the body. Now, let me point this out. Mitosis even forms the organs of the reproductive system. So in the male, these organs that we've just named and the pathways that we have just followed are surrounded by these complete cells. 2N is called diploid because there are 2 sets of chromosomes shown. and every cell in the reproductive system gets them. Now later, after we take this male through puberty, and the male starts actively making sperm, a second process called meiosis is going to occur. And it's going to be in contrast to mitosis, as I said. Cell division, mitosis is equational. Mitosis represents growth and produces all the cells we need to make all our tissues, organs, and organ systems. So in the diploid cells of the reproductive system, and we're doing the male tract, so let's think specifically about producing sperm. We're going to see cell division performed to produce a distinctly different kind of cell. And if you look through the microscope, by the way, I'm an expert at this, and I have spent countless hours looking at chromosomes going through meiosis. So where are we starting? Well, every one of the cells in the body is the same, including the starting cells of meiosis in the gonads. So we're going to start with the same thing. Then we're going to, in this drawing, from here to here, they've actually done two steps. The first step is the same as in mitosis. We copy every one of the chromosomes. So we have 2 black strands and 2 gray strands and 2 red and so forth. However, what's different about meiosis at this first division is that the black and gray, the 2 copies of, I'll call it the number 1 chromosome, pair up. They bond together so that when they line up, it's no longer 2 structures of 2 strands, but it is now a 4-stranded structure. And you'll notice that in terms of colored bodies, there are now no longer 6, there are 1, 2, 3 because of that pairing. Now that means something. This number 1 chromosome, all the strands are now associated into one organelle as we line them up. Now we're going to do something very different from mitosis. In mitosis, we divide, and then we replicate. And then we divide again, and then we replicate. What we're going to do in meiosis from this stage, the first metaphase, is we're going to divide, and then without any replication, we're going to divide again. And that's what produces the reduction. So you will notice that in this division, we took the black, red, and dark blue one way, and we took the gray, pink, and light blue the other. So these 2 daughter cells actually contain the same number of chromosome strands. You'll notice that each side has 2 number 1 strands, 2 blacks over here and 2 grays over here. But one of the things that's really distinct, because the black and gray are homologs, both copies of chromosome number 1, but this came from mom, this came from dad. That means that they carry different alleles. So there are genetic differences between the cells already at the first division. Now, the second division is simply going to separate the strands. So you can see down here, black, red, dark blue are the result of this. And gray, pink, light blue are this result of this product of meiosis 1. So meiosis 1, division of the nucleus, meiosis 2, division of the nucleus, produces these 4 products. Now, if you want to number these chromosomes 1, 2, 3 for black, red, blue, you'll notice that each of these products has 3, half the number, but also has 1 copy of each chromosome type. So there is 1 black or gray chromosome in each cell, 1 dark red or light red, and 1 dark blue or light blue. That's what I mean by a half a set. Now, if you're making sperm out of these, you'll have a 1, a 2, and a 3. If you join that to an egg that has been divided by meiosis, which has a 1 or 2 or 3, what do you get? You get 2 number 1s, 2 number 2s, and 2 number 3s, and you're back to 6. So there's no loss, there's no addition. The sets are exactly balanced. In the 2n, or diploid condition. So here we have the nucleus. This shows the replicated chromosomes paired up and the first division, and then the second division, producing that reduced number. But I want to make this point. Meiosis is cell division to produce gametes. It is reductional, but it is reductional in a very precise way. It's not just half the number. It's an exact half set. Later on, we're going to see examples where those chromosomes are have divided unevenly. Maybe one gets left out of the nucleus and disintegrates, so that instead of having 46 chromosomes, you have 45. Maybe one is replicated and fails to divide at the first division, but divides at the second. Now you have a gamete that carries 24 chromosomes and a fertilization that can produce 47. These always are accompanied by noticeable physical effects, and those noticeable physical effects are detrimental. So I want to make a point about humans. It's not just having a representative of every chromosome. It is having the balance of an exact chromosome number. We'll talk more about inborn errors of metabolism and chromosomal mutations a little later. I should mention Down's syndrome, a syndrome that represents that presents a level of disability to the individual who possesses it, is just such a mutation. Individuals of this type have 47 chromosomes, and the extra chromosome is an extra number 21, one of the very small chromosomes in the chromosome set. And yet it produces an outward appearance of physical traits, and it produces wide-reaching disabilities in a completely viable human being. Now, once you make the gamete, and we're showing the gamete with 3 chromosomes, we're kind of carrying the idea of 6 chromosomes being the total through here. They have to actually change from a cell division product to an active sperm. Think about what that is. A cell division product that's just surrounded by a membrane becomes a packaged, actively swimming sperm cell. The last step, by the way, there's a sequence in spermiogenesis. Genesis means the beginning. This is the beginning of sperm. The last step produces spermatids and a single spermatozoan, or sperm. The process from spermatids to spermatozoa, that maturation process takes 3.5 weeks to make active sperm, but the human male body makes them by the millions. The sperm is a very low-cost cell because it is basically a half nucleus with a motor on it. Organelles that can help its swimming function or its nucleus protection are conserved. Other organelles are reduced. This is a very tiny cell, and one of the problems with our figures is how often they draw the sperm on the same scale as the egg. I mean, actually, if you think about a basketball, if I'm holding a basketball here, and you say that's the human egg, what size would the sperm be? We'll draw it, the size of a baseball or the size of a tennis ball. Well, that's ridiculous. It's more the size of a pellet from a pellet gun. It's very tiny because all it's really doing is packaging this half nucleus right here at the pointer and putting it in a cell that will allow it to propel itself through the female reproductive tract to the location where the egg is going to be released. As a result, this collar right here is very packed with mitochondria. The semen has fructose high sugar. So carbohydrate, from a big portion of the trip, carbohydrate is being absorbed so that ATP can be produced at a high rate. This flagellum, by the way, this is kind of the closest we come to a cell like the microorganisms we looked at. Ciliated and flagellated microorganisms from the pond water. This is an actively moving and swimming cell. Now, the sperm is, as I said, tiny. It consists of this ancient structure, the flagella. You remember it had a 9+2 organization. Here, this base plate does contain centrioles and mitochondria to allow for ATP production. This head contains the nucleus and very little else. Out here we have a pointed tip called the acrosome. It turns out that one problem sperm have is that they come from a body that's different from the female body, so the moment they are introduced, the female body attacks it like a foreign cell. The vaginal opening is an opening to the body, and so like every other opening in every human body, the immune system is there to protect it against unwanted invasion. And that means a lot of mechanisms, the so-called innate immunity mechanisms, that recognize differences and attack them. So here is a sampling of sperm. You'll notice the, in this case, the competent head. The color is well shown here. And then this actually surprisingly long flagella. It swims by waving the base of that sperm back and forth very rapidly. It's a serpentine motion, sending waves down that flagella to push against the water and propel the sperm. This is an actual dissection of the scrotal sac, looking into the testes and epididymis. You can see the kind of spongy nature of the epididymis, and you can also see here the spermatic cord has been opened. The various tissue layers are very obvious. The ductus deferens represents one of the tubes inside this figure. Frontal section through the organs show the erectile tissue of the penis down each side. Now, this is a very, very spongy and porous tissue, one that has continuous, tiny openings. And the secret to the erectile tissue is pumping blood through a one-way valve at high pressure into this structure, and it inflates and eventually hardens this organ. So it goes from a flaccid and limp organ to one that is stiff and can be inserted into the vagina. You see the pathway here, epididymis, ductus deferens, up around, shown behind, looping behind the ureter, behind the central to the urine openings in the bladder, but we're still behind. Down here to this position, where the ejaculatory duct joins the urinary duct. This is where it becomes common. The seminal gland up here, the prostate here, and the vulvourethral, showing its additions to this kind of base of the penis position where it is truly complete semen and is being propelled outward through the penis into the female reproductive tract. Now, we talked about the additions, and I mentioned fructose. Another addition is mucus and lubrication. Makes the semen a little thicker than water. It makes it stick together so that sperm can kind of swim within this semen capsule through a portion of the trip. Another thing that is added is buffers. Buffers based on bicarbonate And the purpose of that is to neutralize the acid conditions that are present as a protective mechanism in the vagina female. So we're going to neutralize, we're going to lubricate, and we're going to nourish the sperm on their trip outside the male organ. Male reproductive functions, how are they developed and how are they regulated? The answer is hormones. Hormones. And the beginning of this begins at the base of the brain in the hypothalamus. Now, this is true for both males and females. There is a general sexual hormone called gonadotropin releasing hormone, meaning that the hypothalamic hormone is going to cause gonadotropin to be released. So when GnRH is released from the hypothalamus, its target tissue is the pituitary, and it's moved there by a portal cyst, straight into the vein, straight to the pituitary, the hypophyseal portal cyst. Very important for the control and regulation of our sexual differentiation, our sexual activity. It's secreted in pulses, and it controls the secretion of the pituitary hormones. Now, these abbreviations, FSH, stands for follicle stimulating hormones, and LH, luteinizing hormone. These are produced in the pituitary, and when GnRH reaches the pituitary, these two are released. Now, I want to point out, up to this point, the names I've used apply to both sexes. So we're going to return now to male reproductive function. And the primary task for male function, I want to point out that males are sexually competent at all times, meaning that they top off those sperm tanks, but they basically can be excited and can deliver their active sperm at any time of their sexual maturity. The big activity in the body outside the pituitary is induced by FSH and LH, and the primary male androgen or sex hormone is testosterone. It is released in response to luteinizing hormone. Now, that means we're born and we grow through our adolescence without the release of these sex hormones. All we're doing is kind of elaborating organs that will have a future reproductive activity. But with puberty, the release of these organs accelerates. It affects the development of the male reproductive appearance as an adult, and it affects the release of higher and higher levels of testosterone. It's the most important of our androgens, and it commences spermatogenesis, or the production of sperm. This process is going on then constantly within the testes. If there is a sexual event, it moves some sperm, 200 to 300 million, out of the testes, and they are immediately replaced by meiosis in the testes. Testosterone also affects the central nervous system, and when I say that, what that means is the male's behavioral traits change. Psychologists and psychiatrists develop a number of concepts of the parts of our mind. And the sexual drive is located in a part called the libido. So the libido, or typical male behavior for reproduction, involves potential mate identification, mate basically approach, mate acquisition, and the actual act of sexual congress. Now, that involves lots of behavioral traits and related activities. When you look at our videos on television about wild animals, you'll often see their sexual behaviors of courtship and of display highlighted. Testosterone stimulates the metabolism of the male body. So the male body does change dramatically. The larynx is changed and the voice typically deepens. Hair growth over the body in male specific areas is enhanced. The shoulders typically broaden along with the increased gain in weight, and a prolonged period of bone growth produces larger and denser bones and more muscle mass. The shoulders are broadened. Now, the pelvic region in the male typically becomes taller, and mineralization of the pelvis makes it a very strong part of the skeleton, but it does not increase disproportionately in width. The testosterone does produce increased blood cell formation and muscle growth as a typical pattern in male development. So the idea, testosterone then feeds back to these hypothalamic centers that control this cycle. So GnRH in the hypothalamus, to FSH and LH in the pituitary, and this is the anterior pituitary, affecting sexual behaviors and sexual drive. The male secondary sexual characteristics are also affected by testosterone. Let me make it real clear. There is in each gender a primary sexual characteristic, and that is, what gamete do you make? If you make sperm, you are a male. It's a physiological definition. If you make an egg, you are a female. But we know from the outside that the differentiation of our bodies at puberty is different in males and females, reflecting their difference in their reproductive role. So, male characteristics that we expect and see, the distribution of facial hair, and especially the growth of facial hair, is produced by testosterone. This increase in muscle mass and body size, and also an increase in the ability to enhance bone density and muscle mass, is an effect of testosterone. Adipose tissue deposits are different in the male. And also testosterone, as the primary sex characteristic, it maintains these accessory glands and their function and the organs of the male reproductive tract. So we've gone through the male system. Now we're ready to look at the female reproductive system and the way that it differs. And we're going to see a dramatic difference. So if we think about an adult male who's sexually active, he's basically making sperm by the tens of millions to hundreds of millions. And that replacement can occur on a basis of hours. The male is basically constantly making reproductive cells. That's a high number, low quality physiology. So let's make hundreds of millions that we use in one act, and that enhances the chance that one sperm will make it. The female reproductive system is very different. It produces an reproductive organ, the ovary, and it identifies potential egg cells within that ovary before the birth of the female fetus. So when that baby girl comes out, her ovaries are formed to the point that all of the cells that can become eggs are already biochemically committed to that path. There's a long period of development, as in the male, but after around 10, 11, 12 years, she becomes reproductively competent. Her entire body will focus on typically the maturation of one follicle every 4 weeks. So compared to the male, in 4 weeks, the male can make, trillions of sperm. In 4 weeks, the mature female matures one egg and presents it for a reproductive event. Female reproductive system is responsible for all the sex hormones and the production of the functional egg. Once fertilized, actually the female body also is responsible for hosting fertilization. And once fertilization is achieved, attaching that fertilized egg and developing embryo to her body and nurturing it through development. And after the development reaches a point where the embryo fetus can become an independently functioning body, the offspring, there's a long period of helplessness. And during that time, the female is equipped with hormone, behavioral, and neurological traits that encourage her fostering, her nourishment, and her devotion to the continued success of that offspring until it can truly live independently of its parents. The organs of the tract begin high up in the pelvis with the ovaries. This is where meiosis is completed and the fertilized egg is expelled from the ovary in a process called ovulation. It is basically thrown out into space and it is vacuumed up into a uterine tube which conducts it to the uterus. Now the uterus is a muscular sac. Normally it is fairly compact and lies without great space requirements in the female pelvis. Once the egg/embryo arrives, it joins with the lining of the uterus and begins its development. Now, if you continue down to the bottom of the uterus and an opening that's called the cervix, passing from the inside out, you would enter the vagina, the opening that connects with the outside, and the external genitalia that are featured on the outside of this external opening in the female body. And we've already looked at this region in detail, and we can see the lower parts here of the reproductive tract, the lower parts of the spine, and this lower sacrum and coccyx arrangement. Fat and muscular attachment are shown here, but basically there are three openings. This is the anus, which is the discharge point for the digestive tract. We have already seen over here the urinary bladder and the urethra and the discharge point for the urinary tract. Sort of between those two cavities, we have the uterus, consisting of layer and layer of muscle, a perimetrium, a myometrium, the muscular layer, notice it's pretty thick, and the endometrium, the lining. This uterus is what will receive the egg. So let's go over that. Up here, this lobed, externally suspended organ is the ovary, where maturation of the egg will occur. This is the open end of the uterine tube. You'll notice those finger-like projections, fimbriae. They can actually move over the surface of the ovary and kind of hover above where an egg will be released. But one key to female fertility is to get that egg into this tube, allow it to move through this tube into the duct that leads to the opening in the uterus. This is where implantation, the attachment to the female body, will occur. This is where gestation, the development of the embryo, will occur, and all the stages of growth and expansion that are necessary for the production of an offspring. Now, where the pointer is, we'll go from being a very, very tiny point where the initial embryo, probably a number of cells that can be counted, burrowing into that endometrium, which has been thickened and biochemically conditioned to receive it. Now, at that point, those cells take off and absolutely just divide, fastest growth in the life of this fertilized egg. And as this happens, this entire structure grows, meaning it adds tissue, but also expands to fill this inner pelvic cavity and push up into the abdominal cavity. Now let's just follow the pathway, because sometimes that egg is not fertilized. It passes down here through the cervix into the vagina, the vaginal opening leading down here to a external opening and to an opening that's protected by two sets of lips, labium minus and labium majus. These tissues have weight at all times, but during the development of the embryo/fetus, that weight is multiplied by a huge number of times. You know, if you think about a 7.5 pound baby being close to normal, the uterine tissue, the endometrial tissue, things like the umbilical cord, the lining of the uterus all grow in concert with the developing embryo. That means structural support. So we do see that the suspension of this female pathway is amply supplied with ligaments. Now, we're in the pelvic bowl, so those ligaments can run up to bony connections and suspend the reproductive tract so all the tubes stay open. The uterine tube runs along the broad ligament, and it opens into the pelvic cavity, lateral to the ovaries. Notice this, opens into the pelvic cavity, lateral to the ovaries. Now, that obviously works very well. There are plenty of human beings. But given the importance of this step, it just seems to me that I would expect to see more certainty in the event. But the open tube allows the end of the uterine tube to actually move over the surface to different positions in the ovary, so that the exact location of ovulation for that month can be found. The mesovarium is a structural support tissue system, including ligaments, that stabilizes the position of each ovary so there truly is a left and right side. Now, we've been going for about an hour, so I'm going to stop this recording for break.